next speaker is Michael Roukes, and the topic is single molecular nanomechanical mass spectrometry towards subcellular and
mechanical elements. So in about 2006 and published in 2008, this technique gave us the ability to essentially weigh the inertial mass. It's sort of mixing terms there, but detect the inertial mass of individual molecules. And the basic technique here is that a molecule comes in and rests on a vibrating mechanical device. I'll show you in the next slide. These are the three guys that were responsible for that. A molecule comes in on a vibrating mechanical device, like a guitar string, think of it, with a resonant frequency shown on the left. And that particular event actually shifts downward the frequency by a slight amount. And we can characterize this frequency shift as a function of the added mass of the individual molecule by something we think of as a gain. Okay. And this gain is very interesting, how it actually scales with the size of the device. Okay, so let me flip this equation around so that I can represent the minimum detectable mass. Now, what's interesting about this is that the minimum detectable inertial mass for these vibrating devices, the ultimate limits, hadn't really been investigated. And so we looked at this for scaling down into these nanoscale devices and convinced ourselves that it would be possible to ultimately detect at the 1 AMU level, so single proton mass. And the reason why this is true is that, as this formula shows on the left, on the right, that this R term, or R to the minus 1 term, which is this gain that I mentioned in the previous plot, depends on how the size of the device in a very strong way. In fact, it depends on this. If you scale the device downward in all dimensions, it depends on this sort of scaling length, or the linear dimension, in inverse fourth power. And so that means from going from microscale to nanoscale, 3 orders of magnitude in size, we get 12 orders of magnitude in this gain. And that turns out to make all the difference if you want to do molecular detection. So just thinking about how devices scale, going from quartz crystal microbalances to SAW or flexural plane wave sensors based on MEMS technology, down to microcantilever devices, down to these nanomechanical devices, it turns out that indeed that 12 orders of magnitude gives you what you need in order to detect individual molecules. So some of our first work back in about 2004 and published in 2006 took some of these nanomechanical devices and exposed them to a molecular beam. And what we saw every time we opened the shutter of the molecular beam is the frequency would shift downwards by, a very measurable or decipherable amount. And if we plot this as a function of the mass added, those were 100 zeptogram, 10 to the minus 19 gram steps. If we plot this frequency shift downward as a function of the mass added, it follows a straight line, and that straight line is the gain. And in fact, we do get this 12 orders of magnitude improvement. We get a mass responsivity of 1 hertz frequency shift for every zeptogram, for every 10 to the minus 21 grams that's added to the nanomechanical device. Now, there's 2 things in this formula, though, for minimum detectable mass that we have to worry about. One is that gain, and the second is the frequency stability of the device, because what we're converting is essentially this added mass into a frequency shift, and there's fluctuations, there's noise that you have to worry about. Now, I could spend the whole time talking to you about noise, and I won't. But just suffice it to say that a lot of work, perhaps the lion's share of work, has gone into really understanding, controlling, and building control systems to actually follow and track in real time and minimize the amount of noise we have in our measurements. And so just going back to this original work in 2006, where we show this 12 orders of magnitude improvement. If we look in this inset here at our noise level, it turns out that the noise level was, for these particular experiments, was about 7 zeptograms, and that's equivalent to 30 xenon atoms. So already in the mass range of looking at proteins. So what's the current state of the art for mass spectrometry? Well, if you translate it in terms of numbers of molecules, and these are different techniques, and this is a few years old, this plot from a book, Siosdak's book, you need something like millions or hundreds of millions of molecules for typical state-of-the-art mass spectrometry techniques. And so we'd like to translate this down into being able to detect individual molecules for reasons that I mentioned shortly. And so what we did is we took these nanomechanical devices and we built them into what one might say is a conventional geometry for electrospray mass spectrometry, where we have an electrospray element at the top that allows us to take proteins in fluid phase, ionize them, and direct them using a hexapole ion column, ion guide, down and deliver them to the nanomechanical devices, at which point they've been denuded of solvents and are just bare protein ions. At the heart is our detector, which is a little nanomechanical device. In this particular case, for the experiments that I mentioned, is a 430 megahertz device. Okay, so what we saw in these experiments was these precipitous jumps downward every time a molecule was added to the device or absorbed onto the device. This is for BSA at 66 kilodaltons. If we add a heavier mass, in this case beta amylase, a couple hundred kilodaltons, we saw bigger jumps. What's interesting is the fact that the jumps were not all the same size. And that's actually a peculiarity of how we're actually doing this mass detection, that it depends not only on the mass of the molecule, but on the position where it lands. So in this original work in 2008, we fired a uniform beam of, or uniform flux of atoms to the nanomechanical device, and we did what we called event probability histogram analysis, which basically says we know what the response function is as a function of position, and even though we can detect the individual nanomechanical landing events, what we're going to do is collect about 500 of them and then use a statistical analysis based on this known position-dependent absorption function. And this was this 2008 paper towards single-molecule nanomechanical mass spectrometry. And I'll just very briefly summarize the results. In the upper left is the position-dependent mass response function translated into a frequency shift mass response function. And what you see is there's two peaks. We call it a bicuspid distribution. And this is what we use, and this gets smeared due to either breadth in the distribution of the analytes or smeared due to the frequency fluctuation noise that I already mentioned. And so what we can do is if we fire a uniform flux of, in this case it was BSA, towards the device, we actually get a mass spectrum that we can then deconvolve using a sum of squares error analysis to get a simple spectrum. And instead of doing it with millions of molecules, in this case, we did it with about 500 molecules. Now the problem with this, of course, is you can't do really complex mixtures, because even for this simple case where we're detecting, 5 different N-mers, of aggregation, states of aggregation for the BSA in our solution, it's just too darn complex to actually try to think about doing this for an arbitrary mixture. And so what we've done recently is a new technique that allows us to analyze molecules one by one as they are absorbed onto the device. And basically what we give up for simplicity in the analysis is we go to a slightly more complex system where simultaneously, in real time, we track 2 for a doubly clamped beam, or for a cantilever, we track 3 modes simultaneously. And the modes shapes shown at the top there, the upper left, are for the first 2 modes for a doubly clamped beam. If you do this, and this is what I was saying, is that you go to a slightly more complex system, where again you have to match to the nanoscale devices with macroscale circuitry, which is always a trick, and to keep the noise down and then track these things in real time at high frequency. After some years, you get it right, and we were able to do what's shown in this particular plot, which at the right side, what you see is the frequency shift as a function of time for different absorption events, and basically the time-correlated jumps. Look at the big ones on the right. The time-correlated jumps between the black and the blue traces for two different modes of the same device, happening at the same time, that's why I say time-correlated, represent a single absorption event, a single molecule coming in and landing on our devices. In this case, it was a gold nanoparticle, not a molecule. We have two equations and basically two unknowns, and so we can solve for the mass and the landing position in real time for this device. I won't go through the error analysis, but the bottom line is we not only understand the mass and the landing position, but we understand the uncertainties associated with those through a moderately complicated statistical analysis, but something that we can do in real time. So the interest now is actually using this for molecular or protein detection. And one of the first things that we did was study IgM, which is known to form isoforms, to exist in isoforms in the blood. This is a picture of the IgM molecule. It's about 200 kilodaltons. And it forms in its sort of most prevalent state in serum. It's an a pentamer. But it has other forms that have immunological significance. And if you have billions of molecules, you can do a page SDS separation of them and actually see the different isoforms that are active in blood serum. Well, what we were able to do is molecule by molecule build up a spectra using this nanomechanical technique. This shows building up a spectrum event after event. Now, if we just represented at each event coming in what was going on, every trace would look like the one I just pointed to, just a single molecule coming in. But in this particular representation, we add them all up. So in the end, we get this sort of white trace at the back that looks like your standard sort of ill-defined spectrum from a complex mixture. But realize that we know the history of how that actually was composed. And so what we can do is, in fact, build up not through numerical or statistical analysis or guessing what the single particle or single isoform distributions look like, but from exact knowledge of the spectrum. Essentially, that's what this technique gives us. So we can represent this in a different way. If you want, we can add all of our individual events together and generate the standard form of a mass spectrum that one would be able to get if one could do mass spectrometry up in this megadalton range. But instead, what this single molecule mass spectrometry does is event by event gives us the exact composition of the mixture that we have. Now, where we're going with this, this is for a single firing a mixture at a single device, which is obviously low efficiency. We have very small capture cross-section. And so what we're doing is using the power of large-scale integration to actually build arrays, detector arrays, pixel arrays that have millions of these mass detectors. And then we'll fire, using microfluidics, contents of individual cells. So this is one particular application. I'll talk about imaging momentarily. So see, these are some of these arrays that we're actually building now in collaboration with Leti and Grenoble, and I'll mention that in a moment. We've already been able to essentially build arrays of 100 devices and then simultaneously read out the results of these individual devices, but we haven't published this work yet. We're always a couple of years behind in what we publish. This large-scale integration is a collaboration between our group and CEA Leti. This is a French Department of Energy, a laboratory for electric and information technology in Grenoble, where they have a $1 billion scale foundry that makes it possible for us to go off axis from standard silicon microelectronics development and use these, create these devices at the nanoscale that move, but do it at the wafer scale. So we have a standard NEMS process that we've developed over the past few years that enables us to build millions of these devices on a chip. And this basically shows our roadmap. Back in 2006, we built the first standard NEMS process at 8-inch wafers. We're now on to our third generation. And this year we'll come out with our, we had last year came out with our first wafer co-integrated NEMS CMOS, and now we're working on the 3D stacking that will be necessary to build these NEMS imaging arrays. Let me end up with my last few slides just sort of presenting the vision for where we want to go with this. One of the interesting applications is, in fact, to do whole bacteria, single bacteria lysates without going to culture, just lysing individual bacteria in order to characterize them. There's a new tool on the market, the Roukes biotyper, that allows you to characterize by the mass spectrum between about 2 and 20 kilodaltons a fingerprint for bacteria, but this still requires culture. So if we can do this from the lysate from a single bacteria in real time, we cut out 2 days typically of the process, which will have big implications for hospital-based detection of MRSAs and other things. So that's an interesting application, but I think the holy grail is to be able to do cell-by-cell mapping of the proteome, and this is a stretch, but I think it's a doable stretch in the next 5 years, that it's possible to think about for a mammalian cell that has typically something like about 1 billion proteins, of mapping all of these proteins by their mass using this nanomechanical technique one by one. If you have, well, I'll skip the arithmetic, but the bottom line is we believe that we can do this in the scale of about 100 seconds or about 2 minutes. Now, we've been focusing using these nanomechanical devices that we have now, given our sensitivity limit of about 10 kilodaltons today, on large proteins. And in fact, our sensitivity goes up as we go to the megadalton range. Our mass resolution, in fact, scales upward as we go up in mass, which is contrary to conventional mass spectrometry techniques. But there's interesting work now that shows, in fact, that our prediction of single Dalton mass spectrometry, this just came out this year, is in fact correct, and that's possible. So over the scale of the next couple of years, we'll be scaling downward in mass resolution. And one really exciting and interesting application would be to actually get a proteomic snapshot at the individual cell level of, for example, the post-translational state of proteins. We don't need single Dalton resolution to do that. As shown here, for a number of the post-translational modifications, 10 or 15 Daltons would be good enough, and that's also good enough to sequence DNA. So our roadmap is shown basically over the next 5 years, we believe will get us to the limit where we can actually do this sort of proteomic fingerprinting at the single molecule level using mass spectrometry. I want to conclude with a couple of comments about imaging. There's beautiful work out of Caprioli's lab at Vanderbilt showing how to use MALDI, matrix-assisted laser desorption ionization, for doing mass spectrometry imaging. And this is really still limited to the macro scale. And in part of the issue is in going, if you wanted to go to the nanoscale and really have subcellular imaging, you would need to be able to handle very, very small amounts of analytes, which is difficult or impossible for conventional mass spectrometry techniques, which of course is the back end of the imaging mass spectrometry technique as we know it now. So what we are proposing to build up is using NSOM, near-field scanning optical microscopy, is an analog of what Caprioli has done, but in fact with resolution, at the nanoscale, sort of subdiffraction limited optics or diffraction limited optics from these nanoscale probes, and using at the back end the nanomechanical array that I've discussed for doing the mass spectrometry. So let me end there. Thanks for your attention. These are some of the great people I've been able to work with to do this.
Efficiency. So you said trying to do this at a single cell level, there are billions of roughly proteins. What fraction of those proteins do you capture in calories?
So today, we can capture a very pathetically small amount. The capture cross-section for the nanoscale device, given the plume, we're doing this by electrospray and MALDI, is really small. So we're at about a part in 10 to the 9 capture efficiency. That's only, though, because we haven't yet applied the nanomechanical arrays. The whole beauty of the technique is that we built it in CMOS-compatible technology. In other words, if you can build one device, you can build millions of devices on the same computer chip. And so we've scaled up to 100 in our first attempt, and our roadmap is actually to have imaging arrays of 1 million within 5 years. When we have imaging arrays of 1 million, then our capture efficiency will be a few percent, which is better than state-of-the-art mass spectrometry, or comparable to state-of-the-art mass spectrometry. So that's a very important point, which you mentioned, but it's something that's really in the realm of engineering challenges rather than fundamental sciences challenges.
So that efficiency, as the way that you described it in the array, limited by the density?
Yeah, the density and the capture and the cross-section of the array, the filling factor of the array. Yes.
You have massively parallel devices like that. Can you reuse them? Can you clean them with ozone, or can you just use them again and again?
So that's a good question as well. The proteins or the analytes that we're actually absorbing onto the device are absorbed onto, we temperature bias the device or the array below ambient temperature, so then the molecules accrete like frost on a window pane, and so we can heat the devices up, and to large part, and not perfectly, we can desorb the molecules. But I want to say that even if the desorption isn't perfect, you know, if you're one by one the molecules are coming in, it takes an incredibly long time to actually build up monolayers of coverage, which is the point where, in fact, the sensitivity is degraded. So the sensitivity, these things have mass dynamic range of, you know, multiple, you know, hundreds of gigadaltons, right? So we can accumulate lots of molecules on the individual devices before we actually have to think about cycling them.
You a unique sample that you've never seen before, how long would it take you to collect the suitable data on mass?
Well, we're not priding ourselves today on the speed of our collection, because for these 73 molecules that we did for IgM, those are accumulated over a few days. And that's because we turn up our flux, but our capture cross-section is a part in 10 to the 9, as I mentioned before, so that means you don't get very many per unit time. So the other question that I frequently get, like this one, is what happens if 2 molecules come in at the same time? And of course, you know, with the capture cross-section and the flux that we have, we don't have to worry about that today. In future, we can adjust the flux. But that's also the answer, going back to your question, the answer of what the rate is, how long it would take us to detect, really depends on the engineering question that was asked of me earlier. And the point is that when we have a few percent efficiency with 1 million detectors assembled by 3-dimensional CMOS and NEMS integration, we project that the limiting time element is how long does it take us to integrate in order to get the requisite amount of sensitivity. And if I fold that all in with 1 million devices, it should take us a few minutes to actually analyze something. So reasonable times on a graduate student lifetime. Thank you.